then product, right? Always remember our goal. Our goal is to digest your polysaccharides through to your individual glucose monosaccharides, which is glucose, through the action of amylase, right? This is our goal. And then for your polypeptides or proteins, to release your amino acids through the action of proteases, which there's a lot of them. And then your lipids or your fats, to release the individual free fatty acids through lipase. And I said all the other substances that we put in our mouth, they're already in their basic form. There's no need for an enzyme to be produced. They just are going to be absorbed. So we put yesterday or Monday, we put the food in our mouth, and right in our mouth, we already come up with this one, right? Because the saliva contains amylase. By the time the food has been processed by our mouth through mastication and mixing with saliva, you have bolus, and the pH of that is already alkaline, okay? And so we swallow it, and swallowing is a coordinated process. Don't forget the cavities that need to be closed to ensure the proper movement or passage of that food. You have to make sure it goes to the pharynx and then the esophagus, and now we go to the stomach. So the stomach is getting an alkaline bolus. The stomach looks like this, and it serves as a mixing chamber, a holding reservoir.
I can't see that. Can you?
This one here?
I can't see the slide number because the
30.
The slide number.
Is that what it is? 30?
Yeah.
Okay, so the stomach, you know, they say if you buy, you know the Italian sausage that we buy, that's the normal size of a typical stomach. But because of the smooth muscle, do you remember the property of plasticity? There have been some stomachs that can extend all the way to the pelvic cavity. And you've seen that in your cat. Some stomachs are small, some stomachs are big. It can expand all the way down. That's because of its job. It's the holding chamber. So look at, when you open up your stomach, so what you need to do now when you open up your digestive organs, in addition to identifying them, I want you to cut them open. So the one thing you will notice about the stomach are the folds. The folds in the stomach are called rugae. Rugae. You will see this word again when we study our urinary system. Rugae refers to or means temporary folds. So when you open up the stomach, I want you to stretch it. Is it true that it can go away? It will disappear. That's the same thing with your urinary bladder. This is different from the folds in your small intestines. That's why you have to cut them all open, okay? In the small intestines, the folds are permanent folds. They're called villi, and it's fuzzy. Like you know the towel has that, what do you call it in the towel? The cloth? So something like this, it's fuzzy for the small intestines. They're called villi or villus. And this one is called rugae. Another very important feature of your stomach is the 3 layers of muscle. Normally, in the entire GI tract, the smooth muscle is just only 2 layers, inner circular, outer longitudinal. This one has an extra, innermost oblique. The reason is the stomach functions in churning. It really, those people, if you're like my Baba in 2 seconds, like I said, he's done eating. So a lot of times, you swallow, you didn't even really chew the food properly. But you need to break it down properly so that the enzymes, by the way, okay, so this is, let's say, my steak. The enzymes can only act on the outside, right? They can't act on the inside. So it will, between steak that's this size and steak that is this size, which one you think will be easier to digest? or breakdown for enzymes. This one, right? Compared to this one. That's why we need to chew. So if you didn't chew your food properly, the stomach will help you with that one. That's why you have that innermost oblique layer. That's called churning. Okay?
Did you say it has 3 layers of muscles
Yeah.
or 4? 'Cause
3.
What is the same 4 layers on your slide? Those aren't muscle layers?
The same four layers: mucosa, submucosa,
Okay.
muscularis.
But it has 3 layers of muscle.
Three, yes, three
Okay.
layers of muscle, smooth muscle.
So what are those things?
What those things?
The mucosa, the submucosa, the nuclearis,
The
what are they?
layers of the wall of, remember?
Oh, the wall, okay.
Of the entire GI tract, remember?
Okay.
This one right here. I said all of them will share the same 4 layers.
Okay.
Okay? But in the stomach, this muscle layer,
The
instead
muscera,
of just
nuclearis,
two,
okay.
there's 3
Muscularis,
of
okay.
them.
Alrighty.
So let's analyze the composition of this
Square.
stomach. If we make a slide of this stomach, so this is how a typical stomach looks like, right? I'm gonna make a slide, and this is how it looks like under the microscope. And it's very easy to identify. I'm gonna prepare slides for you. Stomach, duodenum. I want you to be able to identify or differentiate between the stomach, the duodenum, which is the first part of the small intestines, and the last part of the small intestines, which is the jejunum and the large intestine. So those 4 parts of the GI tract. The esophagus and the pharynx is nothing but stratified squamous epithelium. And you know how stratified squamous looks like, right? Stratified, the topmost is, if it's stratified squamous, the topmost is, squamous is,
Flat?
yes.
Okay.
Don't, do you forget?
No, it's flat.
Did you forget it? Stratified
Things you wanted
squamous, the
something else.
topmost
Yeah.
layer is flat, you remember? So, I'm just going to show you this one. So with the stomach, the one thing, when you make a slide of it, it looks like feathery as far as I'm concerned. You see the difference. It's very feathery. And then in the duodenum, lots and lots of glands, and there's a reason for that one. And then when you get to the ileum, the one thing that's very distinct are the Peyer's patches. You have lots of lymphatic tissue. Then when you get to the colon, lots of mucus secreting glands, so it looks like air bubbles. But let's look at your stomach first. Look at the mucosa. The mucosa is made up of cells. You remember the mucosa is epithelium, and the cells, you call them secretory cells, absorptive cells. You remember that? Enteroendocrine cells. So let's look at the secretory cells, or the different types of cells that make up your mucosa. Okay? Here are the different types of cells that you find in the mucosa. So, mucous neck cell. What do you think will a mucous neck cell make? The reason why it's called neck.
Mucus.
Yes! That's what the mucous neck cell makes, right? Mucus. Because do you know the pH of the stomach?
It's acidic.
Acidic. By the way, if we say acidic pH, is that pH high or low?
Low.
Low. Don't forget that in the test, people always get confused. I say the higher the pH, the more acidic it becomes. True. And you forget that it's opposite, right? So don't forget that. I'm sure you're going to make, one of you will make that mistake. So the higher the pH, okay, the more basic it is. So it's opposite, okay? So mucous neck cells, so one of the cells here make mucus. There is this chief cell. The chief cell is the chief one. That's the one that secretes what you call pepsinogen. This is an enzyme. This is, what do you think is pepsinogen? Is it amylase? No, it doesn't look like amylase. Is it a lipase? No. It's a protease. It digests protein. Okay? The book also says gastric lipase, but not it, the gastric lipase that's being secreted here is not that active. So we're going to compartmentalize the digestion of our nutrients. In the mouth is the beginning of what digestion? Carbohydrate. In the stomach is where we begin protein digestion because of this enzyme pepsinogen. You understand? Okay? Before we proceed, I want you to understand this term, zymogen. Zymogen. Zymogen is what describes all protein digesting enzymes. All protein digesting enzymes are secreted in zymogen form. This refers to the inactive form of your proteases. So all proteases are secreted in zymogen or inactive form. So, okay, do you see this one, this chief cell? I'm just going to make that right here. There's the pepsinogen, right? It's zymogen. So the chief cell made the pepsinogen by protein synthesis, and it's made in zymogen form. Then it's going to release it here, right, into the duct to be finally available here in the gastric lumen. Do you follow? Okay. It is not until it gets here when it becomes activated. The active form of pepsinogen is pepsin. Okay? The reason why it is very important for you to understand that all proteases are made in zymogen form is because if the cell, this is the cell, okay? If this chief cell made the pepsin in active form, it's going to end up digesting itself, right? So they make it in pepsinogen, which is the zymogen form, and then they release it here into the pit, go up here into the lumen, and that is where it gets activated. Okay? What activates this pepsinogen to pepsin is the acid, which is another product that is made by one of the cells here. There is a cell here called the parietal cell. Parietal cell. This parietal cell is the one that secretes, that makes that hydrochloric acid. Now, very important for you to understand that the parietal cell doesn't make pure hydrochloric acid. It's going to harm it too. This is how it makes, this is how it, quote unquote, makes that hydrochloric acid. So this is the parietal cell, you see? So what it releases is hydrogen. It releases them separate, hydrogen and chloride, into the lumen. And when they're in the lumen, these two atoms, that's where they combine, creating an acidic environment. Can you picture that in your head? So this is my parietal cell, secreting hydrogen and chloride into the lumen. And then, see, parietal cell. And then I'm going to write here chief cell. So the chief cell, in the meantime, made the pepsinogen. When the pepsinogen gets mixed with the hydrochloric acid, that's when it becomes pepsin. Are you following? Everybody? You understand, right? So different cells have a different job. Okay? There is another very important secretion from your parietal cell. The parietal cell also secretes intrinsic factor. Intrinsic factor. This intrinsic factor is the one irreplaceable property or capacity of your GI, of your stomach. So people who have had gastric resection or maybe stomach problems, okay, you can synthetically compensate for all of these, like mucus, pepsin. You can help them with this one. You cannot help them with the intrinsic factor. So this, I remember asking that in the test. I forgot how the test bank worded that question. I think, but I remember they had that question in the test bank. Something about the only thing that cannot be replaced as far as the function of the stomach is concerned is this one: secretion of intrinsic or synthesis of intrinsic factor. This intrinsic factor, once it's made and it gets to your large intestines, this is the key for absorption of vitamin B12. B12, okay? And vitamin B12 is very important for the maturation of your red blood cells. And that's why a lot of old people, you know that a lot of old people taking so many medications, it affects the pH of their stomach. It affects their stomachs, period. And a lot of times it affects the secretion of intrinsic factor. This intrinsic factor is mostly concentrated right here in the pylorus section of your stomach, right here. So the stomach, by the way, has different parts. There's your antrum, there's the body, and then there's the pylorus over here. So this pylorus is the common site of peptic ulcers. Now you understand, when you hear someone or when you get a diagnosis of peptic ulcer, you know it's referring to an ulcer in the stomach. And this is the common site of ulcer. But this is also the main site for intrinsic factor formation. So that's why people who have ulcers, a lot of times they become anemic. They're not able to make intrinsic factor, that's the one that's compromised. And so they're not able to absorb vitamin B12, which is the key for forming your RBC. If you go back to that chapter on blood, so they become anemic. You understand? Okay? So anemia can not only be from iron deficiency, it can also be from intrinsic factor deficiency, vitamin B12 deficiency. That's why you hear of some old people going for regular B12 shots or injection. That's to help them make their red blood cell. Okay? So did you notice here in the stomach? Okay, one more thing. In the stomach, acid. That acid is the key to convert your pepsinogen to pepsin. It's the key to activating your protein digesting enzyme. This is one of the reasons why people taking antacids, if they eat protein, it upsets their stomach, because that's where protein is digested, right? So if you're taking antacids, even antihistamines, it affects protein digestion. There's another very important, another nutrient that your pepsin, or no, that the acid targets, the protein in the food that you just ate, right? Acid denatures protein. We're gonna go back to the food that we just digested, the bolus, right? The bolus just contains just chop proteins, remember? What you acted on in your mouth was the carbs, but the protein is still intact. So once that bolus lands in here, it still contains intact proteins. And the acid here is the key to denaturing the protein. So when you denature your steak, it's going to be easier for the pepsin to act on those bonds, the chemical bonds of the protein that you just ate. Are you following? Does it make sense, everybody? Use your flow chart, the one that's in your worksheet. You take that out so you can follow. You understand, everybody? Again, the protein that we just ate consists of lots and lots of amino acids, right? Bound together. This is how they look like. Lots and lots of them. I chewed it. I broke it down into smaller parts, but I still didn't break the bonds. And they are in this form, right? Proteins are always in coiled form. So the acid in my stomach, in addition to converting the pepsinogen to pepsin, it will denature this protein from my steak or my chicken, denature it, uncoil it, revealing this individual linear form of my amino acids. So easy for the pepsin to act and break those chemical bonds. You see that, everybody? Okay? And that's what's going on in the protein. So imagine powerful churning. Powerful churning breaks down the protein into smaller components even further, and then the acid uncoils it so that the pepsin can target the individual chemical bonds, the peptide bonds, okay, of your proteins. Peptide bonds, it's called peptide bonds. That's why it's called pepsin. One more important help that we get from the acid in the stomach is antibacterial, isn't it? That's why when we eat anything contaminated, and they say a lot of times, majority of the times, we ingest possibly contaminated food, maybe harmless bacteria. And the reason they're called harmless is because the acid in our stomach can neutralize them, prevent them from causing any harm. You following? Okay? So, that all of these secretions from these cells is what makes up what you call gastric juice. So when you see this term, you know that it contains mucus, it contains pepsin, it contains all of these. Again, this gastric lipase is not that much. So we're going to compartmentalize again. Mouth is carb, stomach is protein digestion. Okay? Now we get to be introduced to the first enteroendocrine cell. See, once the food lands in the stomach, it's beyond our control already. In the mouth, at least we're still able to control it, right? But once it gets in the stomach, it's autonomic nervous system plus endocrine system. So here's the first endocrine cell that we're going to learn, secreted by the G cell in your stomach. So, like I said, the different organs of the digestive system, they have to talk to one another, remember? They have to know what's going on. Once the food lands here, you have to alert your small intestine. You have to talk to them. You even have to tell this stomach itself to start doing its job. And that's what your autonomic nervous system and your endocrine system does for you. So look at, the G cell secretes this hormone gastrin. And the target organ of gastrin is the stomach itself. What do you think will gastrin tell the stomach? Remember that the food, the bolus is here. What should the stomach do when the bolus is here? Increase activity or no? If the bolus is here, what should this stomach do? It should what?
Bring it down.
Yes, so is that increased activity or no?
Increase.
Increase, so that's what gastrin does. You understand?
Gastrin comes from your G cell.
Of the stomach itself, right?
Okay.
So I'm picturing in my head, once I swallowed, immediately after I swallowed, it's beyond my control. It lands in the stomach. So what tells all these cells? You know, we're studying them, we're studying their job, what they do, but there must be something that's telling them to do it. So you have your nervous system, and the nervous system will target them. Same with this endocrine system. So the enteroendocrine cell in the stomach, called the G cell, secretes gastrin, goes to the stomach and tells it to increase activity. So both nervous system and endocrine system are trying to help one another. Okay? So now we keep uh, breaking down the food, the pepsin acting on the protein. Once once that bolus reaches here, the pylorus, it doesn't look like the bolus anymore. It's actually in liquefied form. And you call that chyme. Okay? And so you see this one, there's a sphincter here, an area of increased muscle tone, and I want you to look for that. You can even see it in the cat. It's so tight, okay? You have to cut it to appreciate that sphincter, but you will feel the tightness there. That sphincter will reflexly relax, right? When there's time that reaches there, it will irritate it. So the receptors will sense that and create a reflex, which is a nervous reflex, to open up, relax the sphincter, allow only a few drops of the chyme each time. So what is the pH of the chyme that's going to be delivered to the small intestine? Acidic or alkaline?
Acidic.
Acidic. Is it high or low? Low. Okay, don't forget that. So now, we're going to put the chyme in the small intestines. So here's your small intestine. This slide, by the way, is very nice. You see, it's just giving you a summary of the events that happened in the stomach. So I'm going to put the food in the small intestine, and you need to know that the small intestine is made up of 3 main parts. The duodenum, the first part is just short. It's called the duodenum. And then the jejunum, which is the longest part. And then the last part of your small intestine is called the ileum. Okay? So of these 3 parts, that's what we're going to compare: stomach, duodenum, and ileum. Okay. Of these 3 parts, which one do you think is going to be exposed the most to a very acidic chyme? The duodenum, right? Because it's the first one that will receive that acidic chyme. So I am thinking that of the 3 parts, do you agree? This must be the one that has to make the most mucus to protect it from the very acidic chyme that it will receive from the stomach. Make sense, everybody? That's how I'm able to identify it under the microscope. So look at the histology of your small intestines right here. See, there is your duodenum over here. What you will notice is this is this is simple columnar epithelium. I don't know if you remember from your histology, remember that? Then you have the goblet cells, remember the goblet cells? Now, in addition to the simple columnar epithelium, we will focus under it. There's a lot of glands here too. Remember, glands are epithelium. So you will see circular glands of epithelium. They're epithelium, and so I know they're glands. These are what you call the Brunner's glands or submucosal glands. So in addition to the goblet cells right here that secrete mucus, this Brunner's glands, submucosal glands right here, they also make mucus. So there's a thick coating of mucus in the duodenum. These glands you will only see in the duodenum. You won't see it in the jejunum, you won't see it in the ileum. That's how I identify the duodenum. You understand? Do you understand why you have those extra glands here, right? They need help, right? Because the pH of the stomach is high or low?
Low.
Low, right? Acidic. So they have to coat themselves with a thick coating of mucus. Okay? So, another and look at, by the time we get to the ileum, I don't know if you see it, but when you get to the ileum, you don't see the glands. Do you see this epithelium?
It's really hard to see.
Okay. Do you see this one? See, there's the duodenum. You see all of this?
Mhm.
This is epithelium too. If you remember so it's also simple columnar epithelium. Remember, I had you draw the simple cuboidal. So maybe simple cuboidal, but there's a space inside. It's you know how epithelium looks like, right? The very cellular, and so they're under this one. So you see, this one makes mucus. This one also they're also glands. So you have a concerted effort between this and this one to make mucus to protect it. Now I want you to look at the ileum. You will see.
Ooh.
Look at the difference now. You see, in the ileum, it's not, you will see this one, very distinct. This is under the mucosa. This is what you call Peyer's patches. These are white blood cells. This is an example of lymphatic tissue. Remember, a tissue is a collection of two or more cells. So you have a lot of white blood cells that uh, congregate together right here in the ileum, because after the ileum is the colon. And the food, actually, you start absorbing it in the jejunum. And so by the time the food lands or gets to the ileum part of your small intestine, this is where feces or poop begins. And we know that feces is dirty, right? There's germs in there. So that's why you will see these Peyer's patches. Again, what is this? White blood cells, lymphatic tissue. They must be here and not in the duodenum. They will be mostly here because this is where feces begins. That's how you differentiate. And you see it under the microscope, you say, oh wow, very easy to identify. Okay? Okay. One more thing we want to appreciate in our duodenum are these circular folds or plicae circularis. So these folds, okay, these folds are permanent folds. They're not the same as the rugae in the stomach. So that's why I said when you open up your small intestine, I want you to cut it open and try to stretch it. See if it will stretch. It won't. It will be fuzzy. It will remain fuzzy. Whereas the stomach, you stretch it, it will become nice and smooth. The purpose of this one, so again, this is permanent. The purpose of this one is to increase the surface area of the small intestine. We need to increase the surface area. Why? Because this is the site where you have to really complete your digestion, number one. And number two, you also need to absorb them. So that's why you need that increased surface area. There's many other factors that help in increasing the surface area of your small intestine. In addition to these permanent folds, okay, your individual cells, you remember? So the permanent folds, you remember the villus? The villus in your small intestine helps to increase surface area. It, when you look at it under the microscope, remember the simple columnar epithelium or like this, right? So that's what's referred to as the villus. You don't see that with your naked eye. And then each and every cell, remember when you were looking up, you're going to focus again. No, I will focus and you will see again. If you, and you will remember that the simple columnar epithelium, that the apical surface, which was a bit hazy like that. It's not cilia. This is called microvilli. So the individual cell membrane, so these are the individual simple columnar cells. The cell membrane is not smooth like that. The cell membrane also have folds. So this is called microvilli. And that also helps. They're permanent to increase the surface area. All of them, they function in increasing the surface area of the small intestine. Again, what is the reason? That's the site where you want to complete digestion plus absorb the nutrients.
Does
And you
it
want to
also
hurry
absorb
up.
the fats?
Yes, you want to hurry up because when you get to the ileum, you're done. You have poop over there. You can't absorb anymore. Okay? So most of this will be found in your duodenum and jejunum area. Okay? So now we're ready to look at what's going on with the chyme that entered my small intestines. The duodenum. So here's your duodenum, right here. Okay? There's your duodenum. And what is the composition of the chyme that landed here? First of all, it's acidic in liquid form. It's acidic. And so this duodenum, you know, the glands, the Brunner's glands that make mucus, all that Brunner's gland does is to protect the cells here. It's not doing anything as far as trying to control this pH. Every part of the GI tract has a specific pH that they work on. For the stomach, it likes to work under acidic conditions. For the small intestines, it needs alkaline. In your mouth, alkaline, right? So again, the glands, remember the submucosal glands, the Brunner's glands, all they do is they make mucus and coat it. You still didn't solve the problem of acidic chyme, okay? And then we still have the intact lipids. There's a few problems here. Then you still have, I'm sure you still have intact carbs and lots of intact proteins still. There will be some glucose, which you digested from the amylase in your mouth. There will be some amino acids from the stomach, but I know there will still be intact proteins, okay? And definitely intact lipids. So we need the problem, we need to neutralize the pH, and we need enzymes to digest all of this. We need to come up with the individual glucose, amino acids, and free fatty acids and absorb it. That's the problem. Okay? What the what the duodenum, the small intestine, secretes are the enzymes that breaks down just the terminal bond. So when you have two glucose molecules bound this way, the small intestine can break that and release. But when it's still big carbs like this, which is always usually the case, the enzymes from your, are you familiar with maltase, lactase, and sucrase? Those are the only enzymes secreted here. It has no enzymes that can no amylase. So amylase is the key to break down these long molecules of sugar, which I'm sure it's still there. Okay? And as far as the proteins are concerned, the only enzyme that's secreted by my small intestine is called enterokinase, which does not digest the protein in your food at all. And it does not even have lipase. This is why your pancreas is so important. All of the enzymes that we need to break down these nutrients to their basic building blocks are from your pancreas. When did we study the pancreas? What body
Endocrine.
system? Endocrine system, very good. Remember, the pancreas makes two hormones, insulin and glucagon. But the main job of your pancreas is digestion. It is mainly an exocrine organ. So I don't know if you remember, but when you were looking at the pancreas under the microscope, there were just a few scattered islets in there. Most of the bulk of the pancreas were the exocrine glands. And what they do is they secrete pancreatic juice is what they secrete. It's called pancreatic juice. Okay? And what is the composition of pancreatic juice? Where's my, right here. See? Look at what the pancreatic juice contains. Look, it contains bicarb. This is the one that will neutralize the acid from the chyme. Again, what do the Brunner's glands, the glands of the duodenum, what do? They just protect the cells. They don't neutralize the acid. What neutralizes it is your pancreatic juice because it contains bicarb. And look at all these enzymes that we get from our pancreas. Pancreatic amylase, which targets what? Amylase targets
Carbs.
carbs. Very good. Pancreatic now you see lipase, which targets?
Lipids.
Yes. And then you see all of these proteolytic enzymes. Okay, very important for you to understand. Remember, they are secreted in zymogen form. You remember? All proteases are secreted in zymogen form. Okay? But the way we activate our zymogens in the small intestines is different from the way they are activated in the stomach. Here, what activates this protein is secreted as trypsinogen. It is secreted in this form and not that form. This is the active form. This is where this enterokinase comes into play. You remember I told you the only enzyme that's secreted for protein, secreted by my small intestine, is this one. Okay? So, this enterokinase, which is secreted by what? My intestinal cells. It's secreted by my intestinal cells. This is the key. It activates trypsinogen, converts it to trypsin. And this one, it's a cascade, will activate the rest. Do you see? I remember there was a question in the test bank somewhere along comparing between the the protein digesting enzymes in the stomach and in the small intestine. So always think there's something different. What is different? In the stomach, you need acid to activate here. Here, it's not acid that will activate your proteases. It is actually this enzyme. Don't forget that in your test, okay?
So
I
in
remember
the stomach, you
there was
need
a question.
acid to activate your enzymes.
Yes, right?
And in the small intestine, you
It's
need
this one.
enterokinase
Yeah,
to activate your
this one.
other enzymes.
Yes.
Okay.
It's not acid. So it's not the same. That's the point. I forget now how they asked that question. But it's not the same. Right? It's not the same because in the stomach it's acid. In here it's an enzyme that activates it. Okay? And then I said this pancreatic amylase is very important because it will act on these large, long chain polysaccharides. The only enzyme that my small intestine has or makes is what you call the maltase, sucrase, and lactase. So maltase breaks down the glucose to glucose bond, right? When they're down to this just two compound, this is where it can act and release. So they can just only target the last two, break down this last chemical bond. That's all they can do. But the long chain ones, this is what is needed, which is from your pancreas. Do you understand, everybody?
You lost me.
Again,
On the maltase, the sucrase, and the lactase, where do they come from?
this is from your small intestine.
Okay.
This one, this one, this one, this one.
Okay.
All from your small intestines, the duodenum and jejunum. Okay? But this maltase acts on glucose and glucose bond, glucose and fructose is what is acted on by sucrase, and glucose and galactose is what's acted on by lactase. Anyway, they can just act on the disaccharides. Once it's down to a two compound form like this can act on it. But if it's still long chain, you need amylase. You understand? Okay? So you know these four enzymes, right? Okay? Okay. I want to look at this fat, the lipid. You remember that fat is insoluble in water, right? We know that fat is insoluble in water. And the main composition of your chyme, it's mainly water. So I'm trying to picture, I will try to picture that fat that landed in my duodenum. I'm trying to picture that fat. And I am thinking right here that fat is in one big glob like this, right? And do you remember I said enzymes can only act on the outside? They can. So there's a problem there. I need to be efficiently digesting and absorb because when you get to the ileum, you're done. This is where your gallbladder comes into play. Okay? Your gallbladder secretes what you call bile. You know what bile is?
Breaks down fat.
That's very good. So bile is a compound which is actually made by your liver. The liver actually makes bile. So look for this in the cat. I'm just going to get the
I do not like this chapter. Way too complicated. With all the enzymes and trying to keep them straight. Oh my
Yeah.
god.
Yeah.
She's going to add nutrition, metabolism, and body temperature regulation on top of that. Oh boy.
That GI tract. Okay. You see your liver? There's your liver, right? You see the gallbladder, you see that green thing there? That's how you identify your gallbladder in the cat also. You feel the gall, sometimes it's not green, but bile is green. But in some cats, maybe it's just, you feel it, it's like a sac. So the liver is hard, and under the liver you feel that cystic thing, it's like a sac. That's the gallbladder, okay? And do you notice how the gallbladder, it's just hanging under the liver, okay? So this one, they just lifted it up to show you. But normally, the gallbladder is hanging down like this. So by gravity, it drips, the bile is made by my liver and it drips down to the gallbladder. So that's why we can't live without a gallbladder. I'm sure you know some people, my mother had her gallbladder taken out. It's not life-threatening to have your gallbladder taken out. But what the gallbladder does is it concentrates that bile. And what is the function of this bile? This bile serves to emulsify the fat. Okay? So what emulsification means is, you know, just like soap, you have small bubbles, right? That's what the bile does. It will break down this big glob of fat into smaller globs. And when it's in smaller globs, it will be more efficient for your lipase to act on it. Do you understand? Right? It's easier and quicker to act on smaller globs than a big glob like this. Because again, enzymes can only act outside. Like if we break down this table into smaller ones, you'd accommodate more people. Right? Okay? And so, the advantage of having a gallbladder is you're able to secrete bile on demand. Like if I had a high-fat meal, I'm going to have more bile being secreted by my gallbladder. If this was taken out, can you picture? It's like it constantly rains the same amount of bile all the time from the liver. You lost the property of concentrating. So whether you had high fat or low fat, it's just the same amount that it secretes. That's why people who have had their gallbladder taken out, if they have a high fat meal, they go to the bathroom right away, right? Because they lost that property of on demand. And as a consequence, the fat exerts an osmotic gradient. They have to go to the bathroom. Does it make sense, everybody? Can you live without the bile? Yes. So the gallbladder, yes. Because it is made by your liver. So you see? So once we get to, by the time you're done with the action of the pancreatic juice plus the gallbladder, the bile secreted by the gallbladder, when you get to this part right here, remember peristalsis? When you get to this part right here, you're already down to your individual basic building blocks, free fatty acids. Now you just absorb. Okay? There is a set of hormones here that helps regulate the activity of your small intestines. These are the enteroendocrine cells of the small intestines. Okay? And there are two important enteroendocrine cells that I want you to remember. You have cholecystokinin and secretin. I I know you're waiting for me to write it down. I'm trying to get something where it is. Anyway, oh, I think I see it. There it is. Okay? Cholecystokinin and secretin. Let's include this one. So once the food gets to the duodenum, in addition to to the mucus secretion, in addition to the enterokinase, the maltase, all of those, the enteroendocrine cells of your duodenum secrete these three hormones. Secretin, look at where it goes. The target organ of secretin is your pancreas. And your liver. See, we know what the pancreas and the liver does, but they have to be told. So this is how the duodenum talks to your pancreas. I need help. This is the one. So what will it tell the pancreas to do? Secrete the pancreatic juice there. That's why it's called secretin. For our purposes, we're just going to concentrate, okay? Partner this with this. Please, for the test to make it easier for you, even in your flow chart, the peanut butter and jelly, okay? And then this one, cholecystokinin, secreted again by the duodenum. You're going to write down that the target organ of this one is your gallbladder. Gallbladder.
These are hormones?
Yes.
Okay.
See, that's how the duodenum tells the gallbladder to secrete bile. It's not an automatic thing. Remember that. They all talk to each other. You understand that everything now talks inside of you. Does it make sense? So in the test, please don't forget to make it easy. And I remember this was asked. I want you to remember that this one will target this one and this one will target. There's another target for cholecystokinin which you need to remember. Stomach. What do you think? Can you guess? If your duodenum is busy, it's busy trying to digest the time that just entered there, right? What do you think will cholecystokinin tell the stomach to do? Increase activity or slow down? That's very good. It will tell the stomach to slow down. Make sense? Because the duodenum is still busy. You want to make sure that you're digesting it, so you can't have the stomach keep on dumping the food in there the time. So this one will tell the stomach to slow down. Okay? Don't forget, target organ for this is this and this. For that one is your pancreas. And this is obvious, right? If I wrote gastric inhibitory peptide, it tells the stomach to reduce gastric activity. Inhibits the stomach. Okay? Break, I want to talk about the liver. Break for 5 minutes, then we talk about